Uninfected MHC cells, MHC molecules bind and transport the antigen fragment to the cell surface. That process is called as antigen presentation. A nearby T cell can then detect the antigen fragment displayed on the cell surface. So depending on their source, the peptide antigens are handled by different classes of MHC molecules. So the class 1 MHC molecules are found in almost all the nucleated cells of the body. They display peptide antigens to the T cells, cytotoxic cells. So class 2 MHC molecules are located mainly on the dendritic cells, macrophages, and the B cells. So these dendritic cells, macrophages, and B cells are also known as antigen presenting cells that displays the antigen to the T cells as well as the helper T cells. But helper T cells mainly binds the the response released which acts on the cytotoxic T cells. This is the structure of the class 1 and class 2 MHC molecule, just a schematic representation. Class 1 and class 2 MHC molecules. The PBR, this is the PBR, alpha 1, alpha 2, and alpha 3, and this is another chain, beta 2. So this is the alpha chain, and this is the beta chain, and it consists of a transmembrane protein. This consists. So this I will explain it later. So this is how it will be present. Class 1 MHC protein, which is for the TC cells, and class 2 for the TH cells. Antigen processing. There are two kinds of antigens, T-independent antigens and T-dependent antigens. T-independent antigens and T-dependent antigens. So T-independent antigens are the large antigen molecules with readily accessible and repeating antigenic determinants. T-independent antigens are large molecules which are readily accessible, repeating antigenic determinants. B cells can bind these directly without being processed. T-independent antigen can bind directly with the B cells without being processed. This stimulates the B cells to differentiate into plasma cell and produce the antibodies. Next, T-dependent antigens. So what you have learned is T-independent antigens. T-independent antigens. So next one is T-dependent. Okay, so T-dependent antigens, these are the smaller antigens with less accessible antigenic determinants. B cells require involvement from the helper T cells to target these antigens. Helper T cells are assisted by leukocytes that process antigens to make the antigenic determinants more accessible as the endogenous antigen. So next, processing of the exogenous antigens. APC, that is antigen presenting cells, first internalizes the invading pathogen and enzymatically digests it into smaller antigenic fragments which are contained within a phagolysosome. The phagolysosome fuses with a vesicle containing MHC2 molecules. Each fragment binds to the antigen binding groove of a complementary MHC2 molecule. The fused vesicle then inserts the MHC2 antigen complex into the cytoplasmic membrane. So the antigen is presented. So the antigen is presented on the outside of the cell. So this is how exogenous antigens are presented. So next, endogenous antigens. So these endogenous antigens are usually the intracellular pathogens are also digested into the smaller antigenic determinants. Each fragment binds to the class 1 molecule located in the endoplasmic reticulum membrane. So the membrane is packaged into a vesicle by Golgi body which is inserted into the cytoplasmic membrane. So the antigen is displayed on the cell surface. So this is how digested by the endocytosis, and in the endosome these are cleaved into the smaller fragments, and then along with the lysosome these are along with the class 2 MHC molecules, these are presented outside the cells. So similarly the class 1 MHC molecules, so how it is internalizes and goes. So these are presented for the TH cells, these are presented for the TC cells. This I think I had already explained in the earlier class. Next, the physiological significance of class summary of the earlier what I said, macrophages. This is the extracellular antigen and it is presented on that. So CD4 helper THS that recognizes the antigen and releases the cytokines. So the effector functions are the macrophage activation, destruction of the phagocytosized antigen. So similarly, this is the B cell. The B cell extracellular antigen binds and the T helper cells, and so that activates the B cells to secrete the antibodies specific for that particular antigen. So this is again the class-sized antigen. So this is this binds to the TC cells. TC cells specific receptor CD8. So but the effector function is lysis. So this is the infected cell for class 1 MHC molecule and the T cell receptor, cytotoxic T cell, and this is the T helper cells which bind and present in the class 2 MHC molecule. So this is the structure of the class 1 MHC molecules, which consists of 2 polypeptide chains, 1 and 2. So 2 polypeptide chains, short beta, and is the cytoplasmic region containing the sites for the phosphorylation and binding to the cytoskeletal elements. So this is the one, and the transmembrane region containing, this is the one cytoplasmic region. This is the transmembrane and highly conserved alpha 3 domain to which CD8 binds. This is the alpha 3 domain. A highly polymorphic peptide binding region formed from the alpha 1 to microglobulin which helps in stabilizing the conformation. Then this is the structure of class 2 MHC molecules, 2 polypeptide chains, alpha and beta. This is alpha and this is beta, are roughly equal length. Both are the roughly equal length, and this the domains are similar to the immunoglobulins. So the cytoplasmic region containing sites for the phosphorylation and the binding to the cytoskeletal elements. This is the one. And next one, transmembrane region containing hydrophobic amino acids, highly conserved, alpha 2 and beta 2. Alpha 2 and beta 2 domains to which CD4 binds. So CD4 binds a highly polymorphic peptide binding region formed from the alpha 1 and the beta 1. So this is about the structure of the class 1 and class 2 MHC molecules and how they are presented and how the antigens are